Welcome to this video on DNA barcoding, rapid species identification using DNA sequences. Let's begin by exploring the core concept, decoding life's diversity. DNA barcoding is a taxonomic method using a short, standardized genetic marker to identify an organism's species. It relies on sequence diversity in targeted gene regions for rapid identification and new species discovery. The supermarket analogy helps illustrate this concept. Like a scanner reading a UPC barcode, DNA barcoding uses a short genetic sequence to identify specimens. Scientists scan the genetic letters and compare them to a reference library for an exact species match. The barcoding workflow involves three main steps. First is sampling, where a minimal piece of tissue is collected. Second is amplification, where the barcode region is isolated and amplified using PCR. Finally, sequencing reveals the genetic code for comparison. Different taxonomic groups have standard barcode regions. For animals, it's the COI gene. For plants, RBCL and MATK genes are combined. For fungi, the ITS region of nuclear ribosomal DNA is used. The Barcode of Life Data System, or BOLD, is the global database for DNA barcoding. It currently holds over 26 million barcode records, allowing researchers to match unknown sequences against a massive, curated library of formally described species. DNA barcoding offers several advantages. It works with fragments, providing rapid and objective identification without needing extensive taxonomic expertise. It also helps detect cryptic species and provides a universal framework for global biodiversity mapping. A key application is in conservation. DNA barcoding is vital in fighting illegal wildlife trade by identifying confiscated animal parts, which provides crucial evidence for prosecuting poachers and monitoring endangered populations globally. Another application is in food safety through fraud detection. DNA barcoding identifies the true species of processed food, combating seafood mislabeling and ensuring restricted or toxic species are kept out of the supply chain. Ecological monitoring uses environmental DNA, or eDNA, which are traces of DNA shed by organisms. By meta-barcoding eDNA from a simple water or soil sample, scientists can catalog an entire ecosystem without physically capturing organisms. Compared to traditional morphology, DNA barcoding is rapid, high throughput, and requires moderate technical expertise. It excels at identifying fragments and distinguishing cryptic species, which are difficult to detect using morphology alone. Regarding species coverage in BOLD, animals, primarily using the COI gene, constitute the largest share at 71%. Plants are at 21%, and fungi and others are at 8%. BOLD currently catalogs over 330,000 formally described species. Challenges remain, including incomplete reference libraries, as many species are unsequenced. Hybridization effects can also lead to misidentification, sometimes requiring complex multi-locus approaches.